the major challenges faced in development of drugs is the process is time-consuming and it is complex, also expensive endeavor with a high rate of failure. To develop a new drug will cost over $2.5 billion on average, and it can take more than a decade to complete. In addition, only 2.01% of drug candidates that enter clinical trials receive regulatory approval. Result from the efforts, only 2.01% of drug development result in a successful marketable drug.
In the pharmaceutical industry, research on small molecules continues because of its multiple advantages. It is easy and inexpensive to make, and many strong, also stable small molecules drugs are already available. Unfortunately, for new small molecule drugs face tough competition from cheaper generic versions. To launch a new drug, it needs a lot of complex data and must run clinical trials, which adds cost and pressure to innovate. Deep learning, DL, is becoming more widely used to predict drug toxicity. By studying large amounts of data on chemical structures and their known toxic effects, DL models can find patterns and connections that might be hard for human experts to spot. This helps researchers identify harmful compounds early on, which can lower the chances of dangerous side effects during clinical trials and make new drugs safer. Using NLP, researchers can quickly find useful information hidden in these texts, such as experimental results, chemical structures, and biological pathways. This helps them discover new drug candidates, identify biomarker targets, and explore effective treatment strategies more efficiently. Some common types include circular fingerprints like extended connectivity fingerprints, ECFP, which focus on substructure patterns, path-based fingerprints that track sequences of chemical bonds, and 3D fingerprints that represent the shape of molecules. GNNs are great at finding patterns in more complex connected data. They work by letting each node share and collect information from its neighbors, helping the model understand both local and overall structures in the graph. This makes GNNs very useful for things like drug discovery, social networks, and recommendation systems.
Reinforcement learning, RL, is a useful method for improving drugs by treating the process like a decision-making task. In this setup, an agent, the RL model, learns what actions to take, like changing a molecule's structure or adjusting dosage, to get the best results, such as high effectiveness and low toxicity. Evolutionary algorithms, which are inspired by natural evolution, are also used to optimize drug molecules. These algorithms work by mimicking processes like mutation and selection, gradually improving molecules over many rounds. Each version is tested based on goals like how well it binds to a target or how drug-like it is. Despite AI's benefits, it also has some limitations, such as the need for large datasets, potential biases, and lack of interpretability. Moreover, AI-based tools should be used in combination with traditional methods to ensure the safety and efficacy of drugs. Lack of transparency, the limitations of AI tools, which transparency makes it difficult to get approval from regulators, since it's hard to prove the model is accurate and trustworthy. Biases in data, a common issue in drug research is that clinical trials often include only certain types of patients. If some groups or conditions are not well represented in the training data, the AI may not predict how well a drug works for those people. Ethical considerations, a major issue is protecting patient privacy, since sensitive health data is often used to train the models. It's important to keep this data safe and secure. Patient information must be collected and used in ways that respect their rights and privacy.